Thank you. I would like to thank the organizer for inviting me to speak to you today. By the way, it's the University of Haifa, not Haifa University. It's the only university it's to separate between us. So I would like to convince you, one moment. Okay. I would like to show it to you today a non-traditional approaches or different model for longevity. And I would like to start with to acknowledge all the guys that support this study, particularly Mariana Andros, a PhD student that just finished her PhD this year, and of course Abraham Giller. צבי, רק תתן לי 3 דקות לפני הסוף. So the problem is that we invest too much time in order to find a model that suits for longevity, and the main problem with longevity, it's something that you can, it's more of a dream that you sell the person because you interact with the people that are 30, 40 years old, and you're talking about a trait, a phenotype that will happen in, let's say, 4 to 5 decades later. So the question, what is the best animal model, so-called, that we can try to study and to follow up and to understand better the biology behind it. Let's start with what we discussed within the 1.5 days since we start, and we're going to discuss it later in this conference. And this is animal model. Most of us use animal model in order to find a trait of interest, the study to understand the trait of interest. And we heard about, worms. We talked about, flies, dogs, mouse, and rats, and even monkeys, okay? However, the problem with this animal model is that they have, well, the pros are they are short lifespan, which means we can appoint an MS student or PhD student or even postdoc, and it will capture the beginning and the end of the life of this creature. And with this, I'm talking about short lifespan, generation time. We can manipulate because this is animal and we can do whatever we want. And we can intervene with the lifestyle of this animal, again, because we can. The problem is they are not human. So the translation between what we study in this animal model, we will always need to take with care, with conscious. It's not all the time translatable. So let's start with human model. And here I'm talking about a disease of accelerate aging. The most known one is the progeria. As you can see, those 2 boys are around 12 to 13 years old, and they're already at the end of their life. This is accelerate aging. There are other syndromes like this, Widerman, and all those 4 that I just listed here. The problem with them is that this is a pathological state. So it's not healthy. So how can we translate what we find there in healthy aging? The generation time is okay because they live short. Manipulation, somewhat we can do, and some intervention we can do. However, remember, those are humans, so we are limited in the way we can do or we can interact with these people. But the good sign on the process, I call it, it's they are human. so the translation is easier. So let's go for the longevity, the guy who lived to 100 plus. So those are the right model to study healthy aging because they are healthy people and they present the trait that we look for. However, they have long lifetimes, so in order to capture what change, you live the same amount of years as they are. Generation time is very long. You cannot do manipulation and you cannot do intervention. However, they are human. So if we take only the concept of those three so-called animal model per se, it shows design, but it's not human model, and there either you can have the pathology or not. Accelerate aging, it's show design, it's human model, but they have the pathology, so you cannot translate it into healthy aging. And longevity, it's not a show design, but it's a human model and doesn't have pathology, meaning healthy aging. So as I said, animal model, accelerate aging, and longevity all have advantage and disadvantage in terms of becoming a realistic, translatable, short-lived human model capable of replicating the aging process in a short period of time. To overcome this obstacle, disadvantage, we propose pregnancy as a translatable, short-term, and natural new human model for aging, which demonstrates several similarities in physiological and cellular degradation mechanism. What I'm saying about this, it's when women become pregnant, we talk about her physiology, not the fetus. Because every trimester, there is progression with the degradation, the physical degradation. Because all the energy, all of the physical physiology of the woman invested in the growth of the fetus. So this takes a toll on the female who carry this fetus. And here what we can do is to follow this degradation over time. And when you look to it, you can find a lot of similarity between pregnancy and aging. So as I said, pregnancy, it's a short design, it's only 9 months. It's a healthy human model, and every woman will agree with me that it's a healthy state. It's not pathological, and as I said, it's healthy. Just to give you a core of similarities between pregnancy and aging, and there is a lot more. We're talking about inflammation. We're talking about accumulation of free radical damage, DNA and protein. We talked about pathological and telomere, which can be a biological marker for aging. And we publish it in aging research. We evaluated similar process between aging and pregnancy by comparing common biomarkers, pathologies, genetic, epigenetic effect to establish the progeny pregnant body as a model for aging. The link is made in terms of current view in aging as byproduct and natural selection, as well as unrelated molecular parallel between the two, relying on existing research and mode of human in other animal when applicable. And last one, we showed that the response of pregnant body to this change and through that illuminate unique areas of potential study to advance our knowledge of the maladies relating to aging and pregnancy and an avenue for solution. So this was a theory. The question, how we go from theory into practice. And this was the next slide. I will show you what we gain. In this scheme, we took three groups: centenarians, offspring, and the control. We used the Affymetrix platform in order to capture the CNVs within those people and to find if there is differences between centenarians, offspring, and control. Offspring is the offspring of the centenarian. Just to capture your interest, copy number variance, it's the same segment that goes within individual, and the change between them can tell you if there's something happened. We next, after we found counted CNV, I mean, we did the analysis, we scanned for CNV that distribute differently between centenarians and control, and we took those candidate CNV and went to mice model in collaboration with Chaim lab. We did quantitative PCR to those guys, meaning the pregnant mice, and we measured the gene expression as well as the DNA copy number. Those were the CNV homologs in mouse model and the adjusted genes, meaning we not just look for the DNA marker, we look for the adjusted genes that are located next to them in order to see if these changes in CNVs affecting the expression of those genes. As you can see, כמה זמן יש לי? Okay, so we saw differences in number between centenarian and controlled blood sample. As you can see, there are 7 of those. They differentially changes. And if we pick on one of them, we can see that there is differential CNV between case and control. Remember, control are 70 years old and centenarians are 100 years old. And we can see that there is a significant difference of CNVs between in this loci between centenarians and control. We look at the gene expression and we saw that there is differential difference. And here I want to draw your attention into this scheme. What you see here, this is what we look for a trend. It's either it's go up with the trimester and after labor it goes down, or the other way around, it goes down with the trimester and go up after delivery, and this counts not just for the CNV, it's also for the gene expression. And the idea is to capture the degradation of those markers with the age of the pregnancy. So what you can see, the CNV trend during pregnancy stage, you can see that within this loci, we see increase and then somewhat decline, and this is more vivid when you look at the expression. Okay? It goes down and goes up after delivery. We saw it in another 8 candidate CNVs, and of course with the genes that adjust into those copy number variants, and you can see there is degradation and increase, degradation and increase, and here it's incline and then decline. So we get trend for both sides. And to finally, what we look for is the epigenetics through methylation. And as you know, the interaction, epigenetic is the interaction between the DNA and the environment, and it can be a result of cosmic effects, surrounding environment, body, organ, tissue, cell, and organelle. And the combination of the environment, the environmental signal and the DNA signal will give you the phenotype of interest. So if we look to it, what you can see, the similarity between aging and pregnancy. You can see that there is decline here. See the level of yellowish until the third trimester, and there is increase after delivery. The same happened here between 50 to 60, and the yellow it's around 80 to 87, but at the age of 95, which means the longevity one, we see an increase in those expression. If you take one of the example, this CNV, you can see that the degradation and then decrease at the age of 95. And if you look at the pregnancy, we can see the same degradation and then increase. With this series of experiments, we propose pregnancy as an improved and more effective aging model that may help us comprehend and complicated feature of healthy lifetime. An additional outcome of this model is that since postpartum recovery age in completion of reversal mechanism, this mechanism may be applicable to aging treatment, delaying the aging phenotype with also reversing aging process, which means if we can translate the end that we saw, the increase of those signatures to aging process, and like in the pregnant lady, she almost recovered to 100%, we can do this effect on aging or partially on aging, and we can decrease the red zone, which is between healthy lifespan and lifespan. Thanks.
Did you by any chance differentiate between first pregnancy or second or third pregnancy? Or the age of the pregnant lady?
It's a good question. We just started, and we wanted to show that this hypothesis works. So when you want to show that the hypothesis works, you take all the green light. Then we can, use more modified models to see if it's working as well.
Okay, thank you.
Thank you very much for a very challenging lecture. And I'm sorry, maybe the question is stupid, but the very straightforward idea. It's well known that pregnancy is associated with immune suppression. Yes, to preserve the fetus is kind of, because it's kind of alien object in the woman body. So can't be, and the same thing occurs with age. Immune system is suppressed. So maybe what you observe is only...
Absolutely, this is exactly what I show. So that with the immune system degradation within aging, it's almost parallel, almost similar to what happened in pregnancy.
Maybe that's what you're studying.
Yeah.
Because it's a very complicated system, a lot of genes involved, a lot of transcription involved.
Absolutely, absolutely.
So it's kind of study of immune deficiency with the age.
You want to do it? I accept.
Hi, thank you for the talk. I always enjoy listening to your talks. I think they're very interesting. I had, guys, two technical questions. One was like, how do you overcome the fact that the data that you're taking from women is longitudinal, but the data that you're getting from, I assume, from the older people is cross-sectional, right? So you're not really following like a guy for 20 or 30 years to see where the genes go. And then the second question is also, pregnant women are under intense metabolic stress, right? Like, I think compared to like ultra-marathoners in terms of like the amount of calories they burn each day. Is there some consideration there also to how that affects gene expression?
So I first answer the second question, which is stress. And I can guarantee you that everyone agreed that aging is stress. So it's not just stress for 9 months, it's stress for 40 years. And for the first one, as it depends how many person you have in this age group. If you have only one per year, it's a problem. But if you have thousands of people that show the same pattern, then it's much more accurate. And also, if you look at and in this case we did it, at a person we looked at them longitudinally for a couple of months, meaning, and we take another sample and we compare and we saw the same trend. Okay? איליה רוצה לדבר.
Thank you. You know, as an historian of aging research, I remember that back in the 1970s, Vladimir Dillman made some parallelism between pregnancy and aging, and he related it to disruption of homeostatic regulation, especially the function of the hypothalamus, kind of elevation of sensitivity threshold. Do you also relate it to this parallelism to central regulation, to the role of the hypothalamus, to sensitivity changes?
It's a good question, and this is exactly what we did in the paper, that I didn't elaborate on all the systems that we compare, but there is much more, and you can, there is a link to our paper in ARR in 2020 that do this comparison with any system, almost any system that we look for. And I think the only difference, which means there was no degradation, it's in the urine system. I'm not really sure about it, but I think this is the only system that's in pregnancy, it's increased rather to decrease compared to aging. But as you said, yeah, there is parallel there as well. No?